Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome to the Deep Dive. We're the place you come to really get under the hood of complex science, regulations, policy debates, the stuff that's actually reshaping healthcare as we speak.
Glad to be here.
Today, we're definitely taking a deep breath, maybe putting on some scuba gear and plunging right into the FDA's world, specifically their very rigorous standards for biosimilar and interchangeable medicines. It can get a bit controversial, too.
Well, it's complexity we need to wade through, honestly.
Yeah.
Because what we're talking about here is, frankly, a revolution in treatment. But it comes with this just staggering price tag. Biologics, these are cutting-edge medicines. Often incredibly complex molecules are absolutely vital for treating serious conditions, and think rheumatoid arthritis, certain cancers, diabetes. But the cost.
Wow. Give us a sense of that cost.
Well, just as an example, a single year of treatment with one of the top-selling biologics, you could easily be looking at upwards of $50,000, maybe more.
$50,000.
Yeah.
Okay. And that expense, that figure right there, is really the engine driving everything we're going to talk about today, isn't it?
Absolutely. It creates this enormous public health need, a pressure for competitive products,
Yeah.
Follow-on versions that can bring the price down.
But there's the challenge, and it's a huge one. How does the FDA guarantee that these follow-on products are just as safe, just as effective as the original drug? Especially given how complex they are inherently, while also making sure there's enough competition in the market.
That's the tightrope walk.
Yeah.
And it's exactly what this sophisticated U.S. FDA regulatory framework is designed to navigate. So our mission for you today is basically twofold. First, define the key product categories you'll hear about: the original reference product, the biosimilar, and the interchangeable biosimilar.
Got it. 3 categories.
And second, walk you through the 2 main approval pathways the FDA uses. These are set up by law, the Biologics Price Competition and Innovation Act, or BPCIA. They're known by their sections in the Public Health Service Act, 351A and 351K.
Okay, 351A and 351K. We'll definitely unpack those. But maybe let's start at the very beginning. Why does this whole separate system even exist for these drugs?
The fundamental reason.
Yeah. The critical concept here, as I understand it, is that biologics are fundamentally different creatures compared to, say, common pills like aspirin or ibuprofen. Those are small molecule drugs.
Exactly. Biologics are often enormous, really intricate molecules. And crucially, they're produced inside living systems, think cells, microorganisms, not synthesized chemically in a lab vat.
Living systems.
That difference, the fact that they come from living cells, not just chemical reactions, that's the single most important factor. It's why their regulation has to be so different and, frankly, so much more complex than for traditional drugs. Okay, so to really grasp the regulatory challenge, you first need a bit more feel for what these biologics are. We're talking about things like therapeutic proteins, recombinant hormones, monoclonal antibodies.
The money mabs you hear about.
Exactly, the mAbs. These molecules can be hundreds, even thousands of times larger than a simple drug like aspirin. And structurally, they're vastly more complex. As we said, they're manufactured in living cells, often genetically engineered cells, using what's called recombinant DNA technology.
And because they are made in these living systems, they have a characteristic that small molecule drugs just don't. You mentioned it earlier, inherent variation.
That's the term, and it's absolutely critical. What it means is even the original drug, the innovator product, isn't perfectly uniform from molecule to molecule or even batch to batch.
Wait, even the original isn't identical to itself all the time?
Correct. There are slight naturally occurring variations. These can exist even within a single manufacturing batch and definitely between different batches or lots, produced over time. It's just the nature of biology.
Okay, so why? Where does this variation come from?
It comes down to how proteins are made and modified in cells. After the cell builds the basic chain of amino acids, it often adds things, modifies things. These are called post-translational modifications.
Post-translational modifications.
Okay. Probably the most famous example, and one that's often clinically very relevant, is glycosylation. That's where the cell attaches these complex sugar chains onto the protein structure.
Glycosylation, I've heard that term. And those sugars aren't just decoration, are they?
Not at all. They can significantly impact how the drug works, how stable it is, even how long it hangs around in your body before being cleared. They're really functional parts of the molecule.
So if a company makes a batch of their biologic this year,
Its glycosylation pattern, the exact types and locations of those sugar chains might be slightly different from a batch they made 5 years ago. Why? Maybe tiny shifts in the nutrients fed to the cells or the temperature in the bioreactor. It's incredibly sensitive.
so subtle changes in manufacturing.
Can lead to subtle differences in the final product. Other variations happen too, like deamidation where an amide group gets snipped off, or oxidation. These are small changes, but they're unavoidable when you're basically using living cells as miniature drug factories.
Okay, so that inherent variation in the original product itself creates a massive scientific hurdle for regulators thinking about generic versions, right? For traditional small molecule generics, the standard is identity.
Precisely. The generic active ingredient has to be shown to be chemically identical to the brand name drug. And then you prove bioequivalence, that it gets absorbed and is available in the body in the same way.
But hang on. If the original biologic drug isn't even perfectly identical to itself over time because of this inherent variation,
Then how can the FDA possibly, scientifically, demand perfect identity from a follow-on product, especially one made by a different company, probably using a different cell line, different manufacturing processes, different controls?
they can't. It's scientifically impossible to guarantee identity.
Exactly. And that realization, that scientific reality, is the pivot point. It forces the regulatory standard to shift. You can't demand identity, so you have to aim for something else.
Similar.
Controlled and acceptable similarity. That becomes the new benchmark. The whole regulatory process, then, is about defining what similar enough means, how to measure it, and how a follow-on manufacturer can demonstrate that their product's pattern and level of variation fits within the same acceptable range that we already see and accept with the original reference product.
Okay, that makes sense. The standard shifts from identical to highly similar within an acceptable range. Now, for you listening, keeping these next definitions straight is really key to understanding how this plays out in policy and at the pharmacy. We have 3 distinct product categories.
Right. All meet high FDA standards for safety and effectiveness, but they differ in how they got approved and what that means practically. First up is the reference product.
The original.
The original, the innovator biologic. This is the one approved through that full standalone biologics license application, the BLA we mentioned. Think Humira, Remicade, Lantus. They did all the massive initial clinical trials to prove safety and efficacy. They set the benchmark.
Okay, reference product, number 1.
Number 2, the biosimilar product. This is the follow-on. To get approved, it has to be proven to be highly similar to the reference product. And crucially, it must show there are no clinically meaningful differences between it and the reference product in terms of safety, purity, and potency.
Highly similar, no clinically meaningful differences. But it's not automatically swappable at the pharmacy, right?
Correct. A biosimilar is still a prescription drug. Your doctor has to specifically prescribe that biosimilar product. It can't just be substituted for the reference product automatically by the pharmacist. Think Inflectra or Amjevita as examples.
Got it. Biosimilar, number 2. And the third category.
This is the highest regulatory tier.
Yeah.
The interchangeable biosimilar product, or often just called an interchangeable. Now, this product first has to meet all the requirements for being a biosimilar.
So highly similar, no clinically meaningful differences.
Yes, plus it has to meet additional, even more stringent requirements laid out in the law. We'll get into those details later. But the key practical difference, the reason this category exists, is about substitution. Where state laws permit it, an interchangeable biosimilar can potentially be automatically substituted for the reference product by a pharmacist without needing the prescriber to write a new prescription. This provides regulatory confidence for that kind of unmonitored switching. Examples here include Semglee and Siltesa.
Okay, interchangeable. Higher bar, allows potential pharmacy-level substitution depending on state law. That's a crucial distinction. But, and this is really important to hammer home before I move on,
Yes.
the FDA's standards ensure that all three categories, the original reference product, the biosimilar, and the interchangeable biosimilar, are considered safe and effective for their approved uses. They all meet the same high bar for patient outcomes. The pathway to approval differs, the substitution rules differ, but the expectation of safety and efficacy is the same.
That's absolutely correct. A critical point for patient confidence.
Okay, so if the science requires this shift from identity to similarity, the law had to catch up to make it happen. You mentioned the BPCIA, the Biologics Price Competition and Innovation Act of 2009. That's the foundation.
That's the legislation, yes. It amended the Public Health Service Act to create these two parallel approval routes we need to discuss, 351A and 351K.
And they reflect really different goals, right? Let's start with the 351A pathway. Who uses this one?
This is the traditional route. It's for the novel, innovator biological products, the reference products we just defined. It requires submitting what's called a standalone biologics license application, or BLA.
Standalone, that word seems important. You're emphasizing the sheer amount of independent work needed here.
Absolutely. Standalone means you have to provide a full, independent demonstration of the product's safety and effectiveness completely from scratch, de novo. This is the gold standard pathway. It typically costs billions of dollars and takes many, many years of research and development.
Billions with a B. Okay. What kind of data goes into that massive standalone BLA package?
It's comprehensive. It has to cover, broadly speaking, 4 key areas. First, product quality, often called CMC chemistry, manufacturing, and controls. Extremely detailed data on how the drug is made, purified, tested, and how consistency is ensured from batch to batch.
Okay, CMC. That's number 1.
Second, animal or non-clinical studies. These are the preclinical studies looking at toxicology, how the drug behaves in biological systems before it ever gets near a human.
Standard preclinical work. Got it.
Third, clinical pharmacology. These are the early studies in humans, often small groups, looking at things like how the drug is absorbed, distributed, metabolized, and excreted pharmacokinetics, or PK, and maybe its initial biological effects, pharmacodynamics, or PD.
PK/PD. We'll come back to those.
Yeah.
And the fourth area, this must be the big one.
It is. The fourth requirement is for large-scale clinical safety and efficacy studies. These are the pivotal trials, often phase 3 trials, involving thousands of patients across multiple sites. Their job is to definitively prove that the product works for the specific diseases or conditions, the indications it's seeking approval for, and that it's safe for that population. This is where that foundational clinical knowledge gets established.
So the 351A pathway, through all that rigorous work, essentially establishes the scientific bedrock. It creates the FDA's prior finding that this specific biologic molecule is safe and effective for certain uses.
Precisely. And without that incredibly expensive pioneering work done under 351A for the reference product, the second pathway, the abbreviated one for biosimilars, simply couldn't exist. It relies on that foundation.
Which brings us neatly to the 351K pathway. This is the abbreviated comparative route. The main policy goal here, as we said, is different. Increase patient access, increase competition, lower costs by making the development process faster and less expensive. How does 351K achieve that without cutting corners on safety?
It leverages that scientific principle of similarity we discussed. The goal for a 351K application is not to independently reprove safety and efficacy all over again. That's already been done for the reference product.
Okay.
The goal is to demonstrate biosimilarity. You do this through a comprehensive, direct, head-to-head comparison between your proposed biosimilar and the already approved reference product. You're essentially showing the FDA, look, our product is highly similar, and any minor differences don't affect safety or how well it works.
So it leverages the FDA's previous finding for the reference product. It doesn't establish it anew.
Exactly. It confirms that the new product behaves in the same way, allowing the FDA to rely on its previous conclusions about the reference drug.
Right, if they're leveraging prior findings, the data needed must look pretty different from a full BLA. What guides the FDA when they're looking at a 351K submission? How do they weigh the evidence?
They use a framework called the totality of the evidence principle. Think of it as being holistic and risk-based. The FDA doesn't just look at one piece of data in isolation. They consider everything together, the analytical data on the molecule's structure and function, any animal studies, the clinical pharmacology data, immunogenicity data, all of it in context.
Totality of the evidence, okay.
Yeah.
And you also mentioned it's stepwise. What does that mean in practice for a company trying to develop a biosimilar?
Stepwise means the development program is designed like a funnel. or maybe a series of gates. You start with the most fundamental comparisons, usually in the lab. The strength and certainty of the evidence you generate at those early stages directly determines what's needed later on.
So the better your early data, the less you might need later.
Exactly. If your analytical comparisons, the lab tests comparing your molecule to the reference product, are incredibly strong, showing very high similarity in structure, function, purity, everything, then the level of residual uncertainty about whether it will behave the same in humans is considered very low.
Residual uncertainty. I like that term.
If that uncertainty is low, the clinical studies needed to resolve it might be quite targeted and relatively small. Maybe just focused PK/PD studies are enough.
But conversely.
Conversely, if the analytical data reveals some subtle differences, even if they seem minor, maybe a slight difference in that glycosylation pattern we talked about, or a bit more of a certain variant, then the residual uncertainty is higher.
Right.
And the FDA will likely require a more substantial clinical data package to definitively prove that those observed differences are not clinically meaningful. Perhaps larger comparative clinical trials become necessary. The required evidence adapts based on the specific scientific findings for that particular product comparison. It's tailored.
Okay, that flexible, stepwise approach, starting with the lab work. It sounds like the foundation of this whole 351K process really rests on that initial analytical comparison.
Absolutely. It's called the Comparative Analytical Assessment, or CAA. And the FDA is crystal clear on this. The CAA is the undisputed foundation. It's considered the most sensitive part of the entire evaluation process for detecting potential differences between the proposed biosimilar and the reference product.
Hang on. More sensitive than a large human clinical trial. That kind of flips the script on how we usually think about drug development, especially for small molecules.
It might seem counterintuitive, I know, but for biologics, it's generally true. Why? Because our understanding of protein structure and function and the tools we have to measure them have become incredibly sophisticated.
Okay.
Things like high-resolution mass spectrometry, advanced chromatography, sophisticated bioassays. These tools can pick up subtle molecular differences, tiny variations in structure or impurities or functional attributes that might not actually translate into an observable difference in clinical outcomes in a typical, say, phase 3 efficacy trial designed to show a drug works.
So the lab tests can see things the clinic might miss.
Potentially, yes, especially very subtle differences. The underlying scientific philosophy is this. If you can demonstrate analytically that your product is highly similar in its structure and its function to the reference product, then it is expected to behave the same way when given to patients. The analytical similarity predicts clinical similarity.
That puts enormous weight on getting that comparative analytical assessment right. What does it actually involve? What are they looking at specifically?
It's an intense, direct head-to-head comparison. The biosimilar developer has to take their product and compare it extensively against multiple different lots or batches of the reference product.
Multiple lots of the reference product? Why multiple?
Because remember that inherent variation. The reference product itself varies slightly from lot to lot. So the biosimilar has to show its own variation falls within the range observed for the reference product. You can't just compare against one snapshot in time.
Okay, makes sense. So what specific features of the molecule are they scrutinizing in this comparison?
They look at a whole range of what are called critical quality attributes. Let's maybe drill down on a couple of key examples. First, as we've touched on, molecular structure. They confirm the amino acid sequence is identical, yes, but they spend a huge amount of effort analyzing those post-translational modifications.
Like the glycosylation we mentioned.
Exactly like glycosylation. They'll use sophisticated methods to map out the types of sugar chains, where they're attached, and the relative abundance of different glycoforms. If the biosimilar's glycosylation profile is significantly different from the reference product's typical range, that raises a flag. It might affect how the drug interacts with receptors or how quickly it's cleared from the body.
And that difference, seen in the lab, could trigger the need for more extensive clinical testing.
Absolutely. It could be the difference between needing only a simple PK study versus needing a larger, more complex comparative clinical study. Another major focus is on purity and variance.
Impurities, like leftover stuff from manufacturing.
Sometimes, but also variants of the protein itself. For instance, proteins, especially large ones, can sometimes clump together, form aggregates. If a biosimilar candidate shows a higher tendency to form aggregates compared to the reference product, that's a concern.
Why are aggregates bad?
Well, besides potentially affecting how the drug works, aggregates can sometimes be more likely to trigger an unwanted immune response in patients. That's a significant safety issue. So purity isn't just about being clean, it's about ensuring the product profile, including variants and aggregates, is highly similar.
And how do they ensure they're catching everything?
The FDA strongly encourages using orthogonal methods. That basically means using two or more completely different analytical techniques to measure the same attribute. If multiple different methods all show similarity, it builds much greater confidence in the result.
Wow, the level of detail at the analytical stage is pretty staggering. So once that strong analytical similarity is hopefully established, then we move into the clinic. But the goal isn't proving efficacy from scratch, right?
Correct. The clinical studies for a biosimilar are designed to confirm that there are no clinically meaningful differences and to address any of that residual uncertainty left over from the analytical comparisons. They're not designed to independently reestablish efficacy.
Okay. So what kind of clinical studies are we talking about? You mentioned PK/PD earlier.
Yes, clinical pharmacology studies are usually the first step in humans. We need to confirm the biosimilar behaves the same way in the body. So pharmacokinetics, or PK, is crucial.
Remind us what PK measures again?
Think of PK as tracking the drug's journey and concentration in the body over time. It measures absorption, distribution, metabolism, and excretion. You need to show that the biosimilar gets into the bloodstream, reaches the target tissues, and stays there for the same amount of time and at the same concentration as the reference product. Basically, that the exposure is the same.
Got it. Exposure and PD.
Pharmacodynamics, or PD, measures a biological or physiological effect of the drug. It's looking at what the drug does to the body. For some biologics, there might be a well-established PD biomarker that's known to be very sensitive and relevant to the drug's clinical effect.
Can you give an example?
A classic example is for drugs like filgrastim, which stimulate white blood cell production. The number of neutrophils, a type of white blood cell, is a very sensitive PD biomarker for filgrastim's effect.
Okay, so if a sensitive PD biomarker like that exists,
Then the biosimilar developer might be able to use what's often called a PK plus PD plus immunogenicity approach. If they show the PK is equivalent and the PD effect via the biomarker is equivalent, that can often provide enough evidence to bridge to the reference product's known efficacy, potentially avoiding the need for large, lengthy, and very expensive comparative efficacy trials.
so that's a key potential efficiency of the 351K pathway, relying on that PK/PD data.
It's a major part of the cost-saving potential, yes. However, for many complex biologics, especially things like monoclonal antibodies used for inflammatory diseases or cancer, there often isn't a single, validated, sensitive PD biomarker that fully captures the overall clinical effect.
So what happens then if there's no good PD marker?
If a suitable PD biomarker isn't available, then the developer typically needs to conduct comparative clinical studies, or CCS. This is often referred to as the PK plus CCS approach.
And what are these comparative clinical studies designed to do? Are they just smaller versions of the original phase 3 trials?
They're not necessarily smaller, but their purpose is different. They aren't designed to prove efficacy de novo. They are designed specifically to be sensitive enough to detect any potential differences in efficacy, safety, or immunogenicity between the biosimilar and the reference product, if such differences exist. They need to be conducted in a patient population where the disease is active and likely to respond to treatment, to maximize the chance of seeing a difference if one is there.
So the goal is confirming sameness, or rather the absence of meaningful difference.
Exactly. And woven throughout all of this clinical evaluation, whether it's PK/PD or CCS, is the assessment of immunogenicity.
Right, the body reacting to the drug as foreign. That seems like a constant concern with biologics.
It absolutely is. Because these are large protein molecules, the human immune system can potentially recognize them as foreign and mount a response, creating anti-drug antibodies, or ADAs.
And ADAs can be bad news.
They can be. In some cases, they might neutralize the drug, making it less effective or completely ineffective. In other cases, they could potentially lead to safety issues like allergic reactions or other immune-related problems. So, a crucial part of the biosimilar evaluation is to show that the proposed biosimilar is not expected to be any more immunogenic than the reference product. You compare the incidence, type, and clinical impact of ADAs between the two products. It's a critical safety check.
Okay, so we've established the bar for biosimilarity. Highly similar, analytically, no clinically meaningful differences confirmed through targeted clinical studies, PK/PD or maybe CCS, and similar immunogenicity. But then there's this higher tier, interchangeability. What's the purpose of this extra designation?
Interchangeability is really about facilitating a specific policy goal, allowing for automatic substitution at the pharmacy level where state laws permit. To provide the regulatory confidence needed for that kind of unmonitored switching, the BPCIA statute set an even higher bar than just biosimilarity.
Higher bar. So an interchangeable product first has to meet all the biosimilarity criteria we just discussed.
Yes, that's the starting point. Highly similar, no clinically meaningful differences. Plus, it has to satisfy two additional specific statutory requirements laid out in the law.
Okay, let's break down those two extra requirements. What's the first one?
The first one is fairly straightforward, conceptually at least. The data must show that the interchangeable product is expected to produce the same clinical result as the reference product in any given patient.
Same clinical result.
Okay.
Seems like that would largely be covered by the biosimilarity assessment, but it's stated explicitly here. What's the second requirement? This is the one that usually involves the switching study, right?
That's correct. The second requirement is specifically focused on the scenario of switching back and forth. It states that for a product that's administered more than once to a patient, which is true for most biologics used for chronic conditions, the risk, in terms of safety or diminished efficacy, of alternating or switching between using the interchangeable product and the reference product must be evaluated. And that risk must be shown to be no greater than the risk of using the reference product without switching.
Okay, so switching back and forth between the interchangeable and the original shouldn't introduce any new risks compared to just staying on the original.
Precisely. And to generate the data needed to satisfy this specific requirement, the FDA generally expects developers seeking an interchangeability designation to conduct a dedicated switching study.
Tell us about the switching study. How is it designed?
Typically, a switching study is designed to mimic a real-world scenario where a patient might be switched between the reference product and the interchangeable biosimilar multiple times over a period. For example, a patient might start on the reference product, switch to the interchangeable, switch back to the reference, and then switch back again to the interchangeable.
Multiple switches. Why so many?
The idea is to assess whether these transitions themselves impact safety or efficacy, particularly looking for any potential increase in immunogenicity, those anti-drug antibodies that might be triggered by switching between slightly different, though highly similar, molecules.
So the switching study provides that extra layer of assurance specifically about the act of switching itself, giving confidence for that automatic, potentially unmonitored substitution at the pharmacy.
That's the regulatory rationale, yes. It's designed to provide the highest level of confidence that substitution is safe and maintains efficacy.
But this is where we hit that policy friction point again, isn't it? We mentioned earlier that some major medical groups are now questioning if this switching study is still scientifically necessary.
That's a really important and evolving policy discussion. Organizations like the American Medical Association, AMA, and the American College of Rheumatology, ACR, which represents specialists who prescribe a lot of these drugs, have publicly stated that they believe the switching study requirement for interchangeability could potentially be removed.
Removed entirely. What's their reasoning?
Their argument stems from the scientific rigor we've already discussed. They contend that if a product has already met the high standard for biosimilarity, meaning it's proven to be highly similar analytically and has shown no clinically meaningful differences in targeted clinical studies, including immunogenicity assessment, then it should inherently be expected to perform the same regardless of switching.
So they're saying the initial biosimilarity assessment, especially the deep analytical comparison, is actually sufficient scientific proof, and the switching study adds little extra value.
That's essentially their position. They argue that the extensive analytical characterization is so sensitive now, and the requirements for demonstrating biosimilarity are so stringent, that the dedicated switching study represents an unnecessary additional hurdle. They see it as adding significant cost and time to development, without providing significant additional safety information beyond what's already known from the biosimilarity data package.
Okay, so if prominent scientific bodies think it might be redundant from a scientific perspective, keeping the requirement seems more about what? Maximum regulatory caution.
You could frame it as maximizing regulatory certainty, yes. The switching study provides direct empirical data on the act of switching, but critics argue this comes at a high price. Requiring these complex and expensive studies can delay the availability of interchangeable options, limit the number of manufacturers willing to pursue interchangeability, and ultimately slow down patient access to potentially lower-cost medicines that could be easily substituted.
So it really highlights that core tension again. Balancing the desire for the absolute highest level of regulatory confidence against the practical goals of timely access and cost savings.
Exactly. It's a debate about whether the current scientific tools are now robust enough that the additional switching study requirement has become more of a barrier than a benefit.
Okay, we've navigated the science, the pathways, the interchangeability debate. Let's talk about how this all translates into the real world for doctors, pharmacists, and patients. First up, safety and identification. How do we keep track of which specific biologic product a patient received, especially if issues arise?
This is critical, especially given that inherent variation in the fact that multiple biosimilars for the same reference product might eventually be on the market, each made slightly differently. Clear identification is paramount for effective pharmacovigilance, so the ongoing monitoring for safety issues after a drug is approved.
And the FDA came up with a specific naming system to help with this, right?
Yes, they implemented a unique, non-proprietary naming convention for all licensed biologics, including reference products, biosimilars, and interchangeables. It consists of two parts.
Okay, what are they?
First, there's the shared core name. This is the official name for the active ingredient molecule itself, like adalimumab for Humira and its biosimilars, or infliximab for Remicade and its biosimilars.
So everyone referencing that molecule uses the same core name. Got it. What's the second part?
The second part is a unique hyphenated four-letter suffix. This suffix is attached to the core name, and it's specifically chosen by the FDA to be devoid of meaning. It doesn't stand for the company name or anything else. It's just a unique identifier for that specific licensed biologic product from that specific manufacturer.
So Remicade is just infliximab, but its biosimilars might be infliximab diab or infliximab adzybda.
Exactly. Inflectra is infliximab diab. Renflex is infliximab abda. Each licensed product gets its own unique suffix.
And that suffix, even though it looks like random letters, is the key tool for safety tracking.
It's absolutely fundamental for pharmacovigilance. If a patient experiences an adverse event, say they develop high levels of anti-drug antibodies or have an unexpected reaction, regulators and clinicians need to know exactly which product they received. Was it the reference product? Was it biosimilar A with suffix X or biosimilar B with suffix KAA? Because even though they are highly similar, they were manufactured using different processes and are distinct licensed products. The suffix allows for precise tracking back to the specific manufacturer and even specific lots if needed. It eliminates ambiguity.
That makes perfect sense. Precision is key for safety monitoring. Now, if someone, a doctor, a pharmacist, a payer, wants the definitive official word on whether a specific product is licensed, and crucially, whether it's designated as interchangeable, where do they look?
The single source of truth is the FDA's Purple Book.
Not the Orange Book. That's for small molecules, right?
Correct. The Orange Book lists traditional drugs and their generic equivalents. The Purple Book is the specific, authoritative database for licensed biological products, including biosimilars and interchangeables.
And why is the Purple Book so important, especially regarding interchangeability?
It's essential because it's the official list that confirms a product's regulatory status, including whether the FDA has designated it as interchangeable with its reference product. State laws that govern automatic pharmacy substitution rely entirely on the Purple Book's designation. A pharmacist needs that official confirmation to know if they are legally permitted to substitute an interchangeable biosimilar for the prescribed reference product. It ensures consistency and safety across the country.
Okay, Purple Book is the go-to resource. Another practical point, product labeling. Does a biosimilar label always look exactly like the reference product's label?
Not necessarily. While the biosimilars label is based heavily on the reference product's label, since it relies on its safety and efficacy findings, there can be differences. The most common reason is what's called a carve-out.
Carve-out, meaning some information is removed.
Yes. A biosimilar might be approved for fewer indications or uses than the reference product. This often happens if the reference product manufacturer still holds some form of marketing exclusivity for a particular indication, maybe a specific pediatric use or a less common condition. Or sometimes the biosimilar company might strategically choose not to seek approval for all the reference product's indications, perhaps to streamline their clinical development program or reduce costs.
So the indications that the biosimilar is not approved for are carved out of its label.
Exactly. For example, Amjevita, a biosimilar to Humira, adalimumab, might have certain adolescent or pediatric inflammatory conditions listed on Humira's label carved out of its own label because it didn't seek approval for those specific uses. So it's crucial for prescribers and pharmacists to always check the specific product label to ensure the biosimilar they're considering is actually FDA approved for the patient's particular condition.
Good point. Always check the label for the specific approved uses. Okay, let's shift gears now to the big picture driver. the economic imperative. We wouldn't have this complex system if cost wasn't a massive issue.
The numbers are stark, and they really tell the story. Biologics, as we mentioned, are incredibly effective, but also incredibly expensive. They currently represent only about 2% of all prescriptions written in the United States.
Just 2%, that's tiny.
Tiny percentage of scripts, but they account for a whopping 40% of total net pharmaceutical spending in the U.S.
40% of the money from 2% of the prescriptions. Wow.
That imbalance is precisely why the BPCIA was created, to introduce competition from biosimilars and hopefully drive down that enormous spending burden on patients, employers, and the government.
And has it worked? Have biosimilars actually saved money?
Yes, they have generated significant savings, though perhaps not as much or as quickly as some initially hoped. Since the first biosimilar launched in 2015, estimates suggest they've saved the U.S. healthcare system cumulatively around $36 billion through 2023.
$36 billion is not insignificant.
Not at all. And the annual savings are growing. Projections for 2024 put the expected savings from biosimilars at over $20 billion for this year alone. So the mechanism can work.
Okay, the science seems solid, the regulatory pathway exists, savings are starting to materialize. So why does it feel like the adoption of biosimilars is still facing so many headwinds? Why isn't it smoother?
Because the science and regulation are only part of the story. The actual market dynamics are incredibly complex and, frankly, often work against the uptake of lower-cost biosimilars. There are some major systemic barriers we need to talk about.
Barriers. Okay, let's unpack them. What's the first big one?
Let's start with payer practices, particularly the role of rebates and pharmacy benefit managers, or PBMs. This often gets called the rebate wall.
Rebate walls. Explain how they work and why they block biosimilars.
Okay, so PBMs manage prescription drug benefits for health insurers, employers, etc. They negotiate with drug manufacturers to decide which drugs get placed on the formulary, the list of covered drugs, and on what tier, which affects patient copays.
Right.
Now, originator biologic manufacturers often offer very large rebates to the PBMs based on the drug's high list price. In exchange for this large rebate, the PBM might agree to keep the originator product in a preferred position on the formulary, and crucially, either exclude the lower list price biosimilar competitors altogether or place them on a less favorable tier with higher patient cost sharing.
Wait, so the PBM gets a bigger kickback from the higher-priced original drug, so they favor it, even if a cheaper biosimilar is available.
That's the perverse incentive, yes. The PBM's revenue might be tied to the size of the rebate, which is calculated off the high list price. Even if the biosimilar has a significantly lower net price after its own smaller rebates, the PBM might make more money by sticking with the originator product that offers the larger gross rebate. This wall of rebates can effectively lock lower-cost biosimilars out of meaningful market access.
That seems counterproductive to the goal of saving money for the system overall.
It's a major point of contention and policy debate. Okay, second barrier, legal challenges, specifically the use of patent thickets.
Patent thickets. Sounds thorny.
They are. Once the main patent on the core biologic molecule expires, the competition isn't necessarily free and clear. Originator companies often proactively build up a dense thicket of dozens, sometimes hundreds, of secondary patents around the original product.
Patents on what? If the main molecule patent expired?
Patents on all sorts of things: specific manufacturing processes, purification methods, formulation ingredients, delivery devices, even methods of use for specific indications. When a biosimilar company gets FDA approval and is ready to launch, the originator company can then sue them for infringing potentially dozens of these secondary patents.
And that leads to
lengthy, complex, and incredibly expensive litigation. This can delay the biosimilar's market entry for years, even if the biosimilar company eventually wins or settles. The sheer cost and uncertainty of fighting through the patent thicket can deter some companies from even trying to develop a biosimilar in the first place. It extends the originator's effective monopoly well beyond the expiration of the core patent.
So rebate walls block access on the payer side, patent thickets block entry on the legal side. What's the third barrier? You called it the biosimilar void.
This is a really concerning trend looking forward. There's a wave of biologics coming off patent soon. Analysis shows about 118 major biologic products are expected to lose their primary exclusivity over the next decade or so. This represents a massive potential market for biosimilar competition, estimated at around $234 billion in potential originator sales.
Okay, huge opportunity for savings. What's the void?
The void is that, according to recent reports and surveys, For 90% of those 118 biologics facing patent expiry, there are currently no biosimilars known to be in development.
90% have no competition lined up. Zero.
That's the alarming figure. It suggests that despite the scientific pathway existing, manufacturers are looking at the combined challenges: the rebate walls making market access difficult, the patent thickets making launch risky and expensive, maybe general uncertainty about pricing and reimbursement, and deciding it's just not worth the massive investment required to develop a biosimilar for many of these products.
So the system designed to create competition might actually be failing to incentivize it for the vast majority of upcoming opportunities.
That's the fear. If this biosimilar void persists, it means patients and the healthcare system will miss out on potentially hundreds of billions of dollars in savings over the next decade simply because the competitive market failed to materialize for most off-patent biologics. It's a potential major failure of the system's economic goals.
Wow. Okay, that paints a challenging picture on the market access front. Let's bring it down to the final crucial piece, the human element. How are clinicians and patients actually feeling about and using these products?
This is where we see some positive signs, but also important nuances. Let's start with physician confidence.
How do doctors feel about biosimilars?
Generally, confidence is quite high, according to surveys. Something like 92% of prescribers report feeling confident in the safety and efficacy of FDA-approved biosimilars. And around 89% say they are comfortable prescribing a biosimilar to a patient who is starting biologic therapy for the first time, what we call treatment naive.
That's encouraging. High confidence in the products themselves.
Yes, and critically, that physician recommendation is consistently found to be the single most important factor influencing a patient's willingness to accept a biosimilar. Doctor trust translates to patient trust.
But there's always a but. What's the hesitation point for physicians?
The hesitation or discomfort really centers on the issue of switching, particularly non-medical switching.
Meaning switching for reasons other than the patient's health.
Exactly. While many physicians are comfortable initiating a switch from a reference product to a biosimilar themselves, in consultation with a stable patient, they express significant discomfort when a switch is mandated by a third party, like an insurance company, purely for cost reasons, without the physician's direct input or clinical rationale.
Why the discomfort, if the products are deemed safe and effective?
Physicians often feel it undermines the patient-physician relationship. They worry about potentially disrupting a patient who is stable and doing well on their current therapy, introducing potential confusion or anxiety for the patient, and potentially impacting adherence if the patient is concerned about the switch. They generally prefer that the decision to switch remains a collaborative one between the doctor and the patient, based on individual circumstances, not just a top-down mandate driven by payer contracts.
Okay, so the issue isn't the drug itself, but the process of switching when it's driven by non-clinical factors. How do patients feel?
Patient concerns mirror some of the physician concerns, but often start from a place of lower awareness. Many patients still aren't very familiar with what biosimilars are.
So education is needed.
Definitely. But among those who are aware, or when the concept is explained, major concerns often revolve around safety. Is this really the same?
Yeah.
And again, overwhelmingly, about the idea of being switched for non-medical reasons.
They don't want to be forced to switch just to save the insurer money.
Patients express a strong desire to be informed about any potential switch, to be involved in the decision-making process, and to have their doctor's input weigh heavily. For someone managing a chronic illness, the consistency and reliability of their medication is paramount. Any change, especially one perceived as being imposed for external reasons, can cause significant anxiety.
So building trust is key for both groups.
Absolutely. And it comes back to education and communication. The FDA's high regulatory standards provide the scientific foundation for trust. But translating that into widespread acceptance requires ongoing education for both clinicians and patients, transparency about the products, and respecting the importance of the patient-physician dialogue, especially around decisions like switching therapies. So when you pull back and look at the whole picture, what we've really seen today is how the FDA framework for biologics and biosimilars operates as this really sophisticated synthesis. It blends advanced molecular science with very pragmatic public policy goals.
Yeah, it starts with confronting that scientific reality. You just can't achieve perfect identity with biologics because of inherent variation. And that forces the creation of this comparative pathway, the 351K, designed specifically to foster competition and lower costs.
While still preserving the rigorous standalone 351A pathway for true innovation, for bringing brand new biologics to patients in the first place.
And the whole system is anchored by the incredible scientific depth of that comparative analytical assessment, which as you said, is often more sensitive than clinical trials in detecting differences. Plus, you have essential tools like the unique four-letter suffix for naming, ensuring we can track safety effectively.
Right. And resources like the Purple Book provide that definitive transparency about licensing and interchangeability status. On paper, the framework successfully balances the need for competition with maintaining incredibly high standards for safety, purity, and potency for all these products: originator, biosimilar, and interchangeable.
On paper, yes. But the challenge, as we spent a lot of time discussing, is translating that scientific confidence and regulatory framework into real-world economic benefits and smooth market access.
Exactly. Those policy hurdles, the patent that gets used in litigation, the rebate walls created by PBM contracts, and that concerning biosimilar void for future off-patent drugs, these are actively undermining the cost-saving potential that the science and the BPCIA legislation were designed to unlock.
Which brings us right back to that fascinating point of friction we talked about regarding interchangeability. Given that major medical societies, the AMA, the ACR, now argue the science is strong enough that the dedicated switching study might be redundant, that it's potentially an unnecessary barrier adding cost and delay.
It really sets up a critical question for the future, and maybe for you, the listener, to think about.
Okay, leave us with that final thought.
How should these regulatory standards continue to evolve? And maybe, how quickly should they evolve? How do we strike the right balance between demanding data that maximizes regulatory confidence and public reassurance versus streamlining the requirements like potentially removing the switching study for interchangeability to realize the maximum economic savings and broadest patient access, especially when the market seems to have these other powerful forces working against those goals already? It's a tough balancing act, with billions of dollars in patient health on the line.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.